It's 2:00. Thank you for joining us today. On behalf of the Ambulatory Care and Endocrinology PRM, I'd like to welcome you to a webinar on the gut microbiome, how it helps and how it hurts. And our speaker today needs no introduction. We have Dr. Kim Kelly with us today to teach us about the microbiome. So thank you so much for joining, and I'll turn it over to Kim.
All right, thank you, Christy. What we're going to do for roughly the next hour is talk a little bit about the gut microbiome. At this point in time, and the amount of information that's out there about the microbiome, we could probably talk for about a year or two. So I'll try to hit the highlights. There are my disclosures. There are the learning objectives for this presentation. Relates to structure and function of the microbiome, especially as it relates to the immune system, and proposed mechanism where the microbiome influences the development of both type 1 and type 2 diabetes. And then finally, the role of the microbiome in comorbid conditions for diabetes, such as cardiovascular risk and depression. So, here's the outline of what we're going to talk about. And I'll start out doing a little ancient history. We'll go back to 1969. How's that? And during that time, a group of 4 musicians, guitarists and singers, appeared at a concert in Bethel, New York, lovingly known as Woodstock. And they became very, very popular. Went out on the concert tour, went out on, started to cut records and so on. And at one point in 1970, right at the beginning of their career, they cut an album called Deja Vu. That's a very famous album for those of you who still have a record player. And the fundamental is on that album was a song that was new to them and new to me. And it's become my favorite over many, many years. And that song is Teach Your Children Well. Bottom line is I'm gonna spare you the agony of me singing it, but I'm gonna start out with the first verse. You who are on the road must have a code that you can live by. And so become yourself, because the past is just a goodbye. And the fundamental here is that we're becoming ourselves literally all the time. We are a pile of cells, human cells, and those human cells are changing over time. At the very least, they're aging. We're painfully aware of that. But the fundamental is those cells are assembled because of things they read in the code that is possessed inside of them. And that's the genetic code. We also have other codes that we can live by. We live by genes that determine both structure and function. The behaviors that we develop, sometimes they're parental lessons, sometimes they're religious lessons, it varies, but we have a number of codes that all of us live by. These codes, all the codes, are influenced by our environment and in ways we are still working to try and understand. So let's start. Humans are a magnificent and complex structure. The average human is a composite of species, a super organism whose outward shell has 37 trillion human cells, approximately 25,000 active genes, and that's an awful lot. Those genes work to assemble those cells into a well-orchestrated pile that looks exactly like us when we look in a mirror. But that's just the beginning. The cells in every person are outnumbered by the bacterial cells in the gut alone by 100 to 1. The genes in every person are outnumbered by the genes in the bacteria 1,000 to 1. And the variety of different organisms that are present in the gut are more diverse than any other colonization outside of the gut. So a very, very diverse population. If you took all the bacteria in the gut and put it all together collectively, that weighs almost 6 pounds. But it's probably one of the most important 6 pounds that we all carry around. So let's talk a little bit about the environment. I'm going to split it into the environment and I'm going to go back one here. Going to split it into the environment, things that we talk about, whether they're weather or pollution or what have you, and the environment, And those are the things that affect us from primarily the gut, but other places within our body. So at this point, let's start with environment. There are a lot of chemicals encountered in modern society that can affect the development of diabetes. For example, arsenic in water supplies, bisphenol A in various forms. All of you have probably had a dose of bisphenol A, at least within the last day or so, from the cash register receipt you handled wherever you purchased something, whether it's a Big Mac or what have you. Bisphenol A coats that paper. And hopefully after handling cash register receipts, you washed your hands before you ate or before you used an alcohol-based cleanser, which would actually enhance bisphenol A penetration into your body. And therefore, this powerful endocrine disruptor chemical is varying a lot of the things that we do and we know about our lives. Finally, you probably all have had at least some emulsifier in processed foods. Carrageenan is a notorious affecter of the microbiome. You see on the left-hand side of that slide, it's red seaweed, bottom line. Carboxymethylcellulose, polysorbate 80. When you're reading the labels on the foods that you get, pretty much anything that you can't read or have trouble pronouncing, that's an emulsifier or a preservative. Whoop. I need to move forward down here. The environment essentially is what we're talking about from the mouth to the rectum, and that's invaded by a variety of organisms that we talked about earlier. And within that system, there are a myriad of absorptive surfaces. You can see here on the right of the slide, lots of the villi are present. Between those are the contents of the gut. And those contents include a whole bunch of things, but primarily bacteria and other organisms. I'm going to have to do them this way. The gut microbiome is made up of bacteria that are primarily thought of as being everywhere from the stomach all the way through the colon. The largest collection of them, obviously, in the colon. 10 to the 11th bacteria per mil of gastrointestinal contents. And the small intestine is really where the action is in most cases as it relates to things that the microbiome do for energy generation. In other words, they help us to metabolize various foodstuffs, generate energy for ourselves, and they generate energy for themselves as well. The microbiome is made up of a variety of different organisms. You see here at the bottom of the right-hand slide panel, all the different phyla of bacteria. Those things change over time. When you're younger, you have more of the firmicute population. And when you are older, those firmicutes eventually begin to give way to have a higher percentage of Bacteroides. But those are the two major families in the gut. That doesn't mean by any stretch that they're the only families that do things, as we'll see as we move through the talk. Many things affect the type of bacteria in the gut. First and foremost, when you are born, as if you are lucky enough to pass through a vaginal canal, you acquire a number of different bacteria from that passage. Not sure why that went that way. And as you acquire those bacteria, they're then present in your GI tract and begin to help you by making energy, by metabolizing a variety of things, including human milk, when you are beginning to nurse. Cesarean section is not associated with those types of populations of bacteria. And so from a practical standpoint, you are already at a disservice by not having had a vaginal birth. Once things get going, you are colonized by a variety of bacteria, and that colonization changes over time, depending upon feeding method, and also depending upon whether human milk is one of the foods that you ingest over time. Add to that environmental exposures and so on, and eventually as you become a toddler, your microbiome tends to stabilize, I guess is the best way of saying it, coming closer and closer to what you will have in your adult life. A very famous gentleman, and I think all of you will recognize this quote, "Tell me what you eat and I will tell you what you are." Your microbiome is a reflection in many cases of what you eat, along with bacteria that you inherited, genes that determine some of the bacteria you will engraft in your GI tract, and so on. So food is a major, major component of the microbiome, dictating almost exclusively the content of any one individual, being somewhat different than the content related to any other individual. So here's a great graphic of an exposure. These were infants whose gut bacteria were tested after a few days. These are days along the bottom. And you'll see that when you're ingesting breast milk, you ingest not only lactobacillus and bifidobacteria, but a whole host of others, beginning almost in the second week. As you move through time, ingesting rice cereal, then table foods, then eventually an adult diet, the development of very, very diverse populations of bacteria are the goal here. And so the more diverse your bacterial population in the microbiome, the better off you are. One of the things you can see here in this one individual who was studied is that they received antibacterial agents, which very quickly changed the population of bacteria pretty close to what it was whenever you started your life. So we all know that antibiotics dramatically change the flora of the microbiome, and it does take a while to recover. Gut bacteria help to provide energy. They break down carbohydrates. A good example here is an organism called Bacteroides cellulositicus. 76 different carbohydrate active enzyme families present in just this one organism. 56 of those are not represented in the human genome. So you have a population of bacteria that are really helping you by breaking down things that your normal genes would not be able to help to break down. In other words, breaking down things to supply the nutrients for things that you're going to do. But they do a lot more than just help you to generate and absorb energy. So let's talk a little bit about the immune system. And we're going to start by showing you kind of a graphic here, cartoon of where this is in the small intestine. We're going to talk about two basic areas. First, how you are acquiring the antigens and defining whether or not these are antigens you should react to or not. And then the cellular system that keeps this barrier constant or keeps it tight. So we're going to talk about acquisition of antigens through segmented filamentous bacteria. And these particular cases, whoops, I didn't do that. These bacteria high in the ileum are present in the contents of the ileum, and they're sampling bacteria and other things, other food antigens as well. Then they bind to the epithelial cells, and these antigens are actually brought into the lamina propria, which is in turn an area primarily set up to start understanding and recognizing immune things that you need to react to and things that you don't need to react to that are simply commensals. Here's a microgram of segmented filamentous bacteria. You can see in the center panel, there's actually a bubble formed, and that bubble is trapping the antigens that bacteria has brought into the epithelial cell. Those antigens then are passed through to the lamina propria. They stimulate the Th17 cells. They generate a modest reaction so that your system remembers that. The makeup of the gut microbiome can change quite rapidly. In a matter of days, when the diet is changed from primarily plant-based to primarily animal-based, the changes in fiber, fat, protein, and in different bacteria thrived, and others tend to make a back seat. Just whenever the nutrient population changes, your body is accommodating to it, or more appropriately, your microbiome is accommodating to it. Tight junctions, those are the things that hold us together, actually all of our cells pretty much, but very important in the gut, in the epithelial layer, because there's a lot of not so nice things that float in the contents of the gut. And we would like those not so nice things not to leak in to the bloodstream, or not to leak into the lymphatic system, or what have you. And so we keep these junctions between cells pretty tight through a variety of different things. At the bottom left, you'll see the panel talking about things that can disrupt those junctions. And I want to point out to you at the right-hand part of that panel that viruses play a significant role in the ability to cause separation of various cells by attacking different components of the process that keeps the cells connected together. In humans, there is an additional chemical or additional substance by the name of zonulin that when it stimulates a certain receptor, when it is released, and we'll talk about things that release it, when it's released, it activates its receptor, and that receptor, through a cascade of reactions, causes an actin and myosin filament to begin to contract, and that contraction literally pulls one cell from the other. So zonulin is the only endogenous component that we know of currently that can actually stimulate that process. Vitamin D plays a role in the synthesis of many of these. But here it's important to recognize that for your immune system and for the maintenance of that, vitamin D is very, very, very important. The immune system then determines what things are commensal and what things don't need to be changed, but basically need to be remembered, especially something that has the likelihood of hurting you. So there's a memory system involved with this through the T cells, and a system of reaction through the B cells to produce antibodies and so on to the things that are not to be tolerated. When you talk about the microbiome or microbiota, B's are the key. That's a good way to remember it. First B is bifidobacterium. 90% of colon microbes in neonates delivered vaginally are bifidobacterium, and there are many reasons for that, but those particular organisms have a great role to play in the metabolism of a lot of different substances. They degrade non-digestible carbohydrate, which is important. We'll show you why that is in a minute. They protect against pathogens. They produce vitamins. They also stimulate the immune system and have a role in neurodevelopment, primarily in helping with the Bacteroides organism development too, which is also pivotal to neurodevelopment. They enhance butyrate production in anaerobic butyrogenic bacteria, which is very, very important, as we'll see in a bit. Bifidobacteria, the best way to think of those are microbiome food processors. They feed on a whole host of different substances, plant-based fructans, polyglycosides, you name it, and they begin to break down these substances into other substances that other strains of bacteria can use to produce things that they are responsible for, such as butyrate, propionate, acetate, and so on. So there's a developed cross-feeding system between different strains of bifidobacteria and different strains of other bacteria as well to produce some of the things that are important in our immune system and in maintaining the epithelial barrier in our gut system. The other B is butyrate, which we were just talking about. Butyrate stimulates mucin secretion, so it increases the gut microbiome or the gut barrier to having organisms cross that barrier. They increase mitochondrial function and biogenesis, so they stimulate the epithelial cells to generate energy primarily through the TCA cycle. They also fortify tight junction proteins, so they're kind of a good glue that helps to hold the tight junctions together. They influence the immune balance between helper T cells and regulatory T cells, and they are responsible for stimulating the release of gut hormones. Butyrate is really the workhorse of the microbiome. It has a number of different functions. First of all, it increases mucin gene, which you can see here in this little insert, increases the activity of the mucin gene to stimulate the production of mucin, and also stimulates better adhesion and the synthesis of adhesive components of the tight junctions. It also stimulates the release of hormones such as GLP-1 and PYY. And a very important function of butyrate is that it inhibits class 1 and 2 histone deacetylases, reducing the production of inflammatory mediators. So it is an anti-inflammatory for sure, and it also helps to differentiate T cells into regulatory T cells, and therefore reduces the presence of inflammatory cytokines. Microbiome soul food is really short chain fatty acids that come from the metabolism and breakdowns, probably started by a number of different bacteria and ended with the so-called butyrogenic bacteria. And those are the ones that release acetate, that release propionate, and release butyrate. The short chain fatty acids are really where the action is here, butyrate being the most important one. In the healthy gut, there's a fermentation of dietary fiber that that produces short chain fatty acids. And then the butyrate producers, Clostridia clusters, and a couple of them that you see listed on this slide, those then take the short chain fatty acids and use them as energy. And in addition to that, butyrate is oxidized here in the cell, the second insert you see on the right, to acetyl-CoA, which is the main source of energy for the colonocytes. So we've got some structure and function down. Let's talk a little bit about some roles in disease. The role of disease is a really interesting one. The current state of the art in microbiome disease relationships is something I like to call guilt by association. That is, we pick a cohort of individuals with a given disease, then we study their microbiome. Then we begin to study whether the microbiome pattern is causing the disease or is a byproduct of the disease. And once we discover or feel our way through that, then if we believe it's causative, then we start to go down through the mechanisms by which it caused the disease. And while we know a number of them, we most assuredly do not know all of them. So this is primarily right now guilt by association. And what you'll see in the literature is in this particular disease, there was a decrease in firmicutes and an increase in Bacteroides, or this and that ratios were changed. So fundamentally, we have a good feeling of what the pattern is. We're trying to look now for why that pattern is associated with the disease. What is causing that particular disease or affliction? The relationships of the gut microbiota are a whole different variety of places. One of the ones that become quite important in a lot of different diseases are genes and genetic relationships. While there is some heritability of the gut microbiome, about anywhere from 5 to 45% of your individual variation in the microbiome is explained by your genetics. That could be smaller in some patients, it could be larger in other patients, depending upon what genes you have. Host genes are as components along with the microbiome are probably the place where a lot of disease relationships come into play. Those host genes that are components are often associated with the major histocompatibility complex. Can't say that fast. MCH2 and to some extent MCH1 is involved in many diseases with microbiome actors involved in the pathophysiology. Genome-wide association studies and whole genome sequencing have uncovered over 70 SNPs associated with variations in microbiome profile and microbial abundance as well. So you are all familiar with the major histocompatibility complex, the chromosome array on chromosome 6, There are the class 2 histocompatibility antigens, HLA-DR, DQ, and DP. Many of the microbiome components associated with disease also have some relationship with these components, and we'll talk about those in a little bit more detail in a minute. So some mechanisms of disease. This is absolutely not comprehensive. I'm just using some examples. The HLA-DR3 and 4, HLA-DQ2 and 8, they are associated with type 1 diabetes, and if present in the right blend, also associated with concomitant celiac disease. Multiple sclerosis has a whole different system, usually associated with Epstein-Barr virus. Ankylosing spondylitis, yet a different relationship associated with a significant decrease in lactobacillus and therefore an increase in Th17 hyperimmunity. Tumorigenesis and chemotherapy resistance are associated with Helicobacter pylori, which is a major nasty player in the gastrointestinal tract, associated with gastric ulcer, gastric cancer, pancreas cancer, esophagus cancer, you name it. Fusobacterium nucleatum, a common finding in colon cancer and also in chemotherapy resistance of treated colon cancer. Dysbiosis in bacterial products, inflammatory bowel diseases such as Crohn's, UC, IBS, inflammation such as the leaky gut and type 1 and type 2 diabetes, which we're going to talk about in a little more detail. Gut bacteria metabolites that cause some pathologic changes, including trimethylamine, imidazole propionate, and a decrease in the production of butyrate, which leads to insulin resistance in type 2 diabetes, especially in the presence of imidazole propionate. Those are just a couple of examples. I could go slide after slide to detail more, but these give you some examples of the variation in the ways in which the microbiome participate in disease. Dysbiosis. This is one of my very favorite. I love the term dysbiosis, which assumes by definition that there's a eubiosis of some kind. What dysbiosis really is, and I've chosen to come up with my own definition, and that's more of the bacteria that do bad things and less of the bacteria that do good things. And that encompasses a heck of a lot, but your gut bacteria, the microbiome patterns change. And when those patterns change from what they are in you normally, we call that dysbiosis. There we go. Yeah, there we go. I'm sorry. The intestinal barrier, where all of this is, where the action is happening. In lean individuals, that intestinal barrier is quite functional. You can see here the dark blue essentially is a very thick mucus that coats the epithelial cells. Through that mucus and through those cells are presented antigens, and those antigens help you to develop immune responses, either tolerance or immune reaction to those particular things that finally are leaked into the lamina propria. In individuals with so-called dysbiosis, usually associated with obesity, there is a higher population of the bacteria that do not so good things, the red ones, and a decrease in the barrier that the mucus provides, and then also a decrease in the barrier that the junctions between cells would provide. So at this point, you have bacteria that are closer to these cells. You have bacteria that can pass between cells, or pieces of bacteria, lipopolysaccharides and so on, can also translocate into the area where you're developing immunity and developing whether or not you're going to react to that particular pathologic change. One of the things we do know in individuals who eat a so-called Western diet is that there is a relative decrease in the mucus content, the mucus barrier. And as a result, the bacteria, here you see on the left, that's a normal person. And you can see in the green, there are 2 yellow bacteria associated, and there's some distance from the epithelial cell. But whenever you're eating a Western diet and a whole bunch of other changes are happening, which we're going to talk about, what you see is that there's a thinning of the mucus barrier, and thus these bacteria are much closer to the epithelial cells and much more likely to pass between the cells or be detected by the dendritic cells and so on to generate an immune reaction. In the upper right-hand panel, you'll see some distance from the epithelial cell to the first bacterium, and you'll see it's quite dramatically lower in people with diabetes than it is with a normal person. And when we talk about the so-called Western diet, I get a big kick out of it. Everybody assumes that we feed mice fat, and when we feed mice fat, bottom line is that's generating something that looks like the Western diet, or generating changes in the gut that looks like the Western diet. But the Western diet actually contains not only high fat, but high sugar and high sodium, and a whole variety of things. For example, some of the emulsifiers that you saw right in the very early slides, all of those are present in the Western diet. So be careful when people talk about a Western diet being reproduced by giving mice a high-fat diet. That's not exactly synonymous with what you see in human beings eating a true Western diet. There are many different causes for type 1 diabetes that have been proposed. Years ago, cow's milk was one of those. We have some pretty decent evidence that viral infections, especially with an enterovirus family called Coxsackie B, those things are associated with the development of type 1 diabetes. And we have some more recent evidence that the leaky gut, which is present in people who are going to develop type 1 diabetes, that leaky gut can let things leak in that can then also mimic some of the proteins that you find in the pancreas, and therefore you develop an autoimmune reaction. Whoops, take that. I apologize, this should not have gone forward. Essentially, some of the bacterial products can then cause an autoimmune, result in an autoimmune destruction of the beta cells. And that's what we're going to dive into here. In type 1 diabetes, like I say, we've moved from viruses being the primary cause. Now we have evidence for bacteria being a contributor. And my bias and the bias of a number of articles essentially say that it's probably a combination of those. We do know that enteroviral infections can predate the development of immunity, the development of antibodies to a variety of different things. And as a result, it was thought that once the enterovirus is in you and persists in you, that's what's causing you to react to your pancreas. And there are demonstrations of enterovirus actually being present in the pancreas. However, in addition to that, we also know that the relationship between let's see if I go back here one. We also know that the relationship here between the histocompatibility antigens is quite prominent. So why is it that HLA-DR3 and HLA-DR4 are more associated with it? What does that have to do with a virus? Well, interestingly enough, not much. However, we do know that there's binding sites on T cells and binding sites on the major histocompatibility antigens that actually look the same. And so the current working theory, and I would suggest on this slide, there are references, the reference at the bottom, the Morse and Horowitz article, suggests that what you're actually doing is the viruses are opening up the junctions between cells, bacteria, especially Bacteroides, and some of its breakdown products leak in. Those then bind to the major histocompatibility complex, and then in turn, they are action sites for the development and action by T cells to react to something that looks like a peptide from an insulin peptide, but actually is a bacterial peptide. So it may be that these potential causes are actually working in tandem. Genes linked to type 2 diabetes, my God, there are many, many hundreds of them. About 15 of them are consistent from study to study. In general, the general population has about a 33% lifetime risk for type 2 diabetes. If you have a specific SNP TCF7L2, that increases your risk of lifetime diabetes to over 60% in many cases. There is a cumulative effect of different type 2 diabetes linked genes on diabetes development. If any of you have had your genetic profile done, and I have looked at what those genes are reported to be and looked at what my likelihood is of the 15 consistent genes associated with type 2. I actually have 5 of them in my genome. So it's a wake-up call for sure, and would certainly suggest any of you to have your genome done. In type 2 diabetes, once the inflammation occurs in the lamina propria with T cells and the release of inflammatory mediators, then essentially those compounds decrease liver insulin sensitivity. Some of the bacteria stimulate the production from histidine of imidazole propionate, which also cuts down on the sensitivity to insulin and in its release as well. So at this point in time, a lot of the stuff from the inflamed cells, whether they're fatty acids or what have you, cut down on insulin effectiveness and actually the release of insulin as well. The leaky gut leads also to systemic inflammation. And this is one that I really want to emphasize as a thinking point here. We talk about the leaky gut allowing bacterial products to leak into the lamina propria, and you develop potentially antibodies to them, or you develop T cell reactions to them. But once inflammation sets in, then what you do, you have an endogenous release of that substance we talked about very early, and that is zonulin. Plasma levels of zonulin increase in a whole variety of different potential pathologic problems. For all intents and purposes, you'll see that they have many, many different functions within the immune system. And all of these zonulin increases the separation between cells pretty much everywhere. We talk about it in the gut in order to talk about some of the mechanisms of the microbiome. But in addition to that, they actually decrease the effectiveness of the blood-brain barrier, and they have been associated with a number of diseases that have gone actually to brain inflammation, such as Alzheimer's disease, Parkinson's disease, and so on. So a leaky gut isn't just the gut. The leaky gut is actually the leaky person, and the leaky part of the gut problem actually leads to more pathologic changes in other tissues. Finally, let's talk about what the gut microbiome organisms do to contribute to CV risk. Cardiovascular risk is obviously significantly increased in individuals who have diabetes, also type 1 and type 2. We know, and this slide's probably been in every presentation you've ever encountered as we talk about lipid toxicity, and that is the bacteria in the gut produce metabolized choline, and that choline can be converted to trimethylamine, which is then oxidized in the liver to trimethylamine oxide. And one of the things we know is that nearly all of the bacteria in the gut actually have the enzyme to convert choline to TMAO. So it's not something where we could just get rid of one bacteria. Choline is ubiquitous in our food, and almost all the organisms possess the ability to take choline and make it into trimethylamine. However, carnitine, while present in red meat, and therefore one of the prescriptions for cutting down on the toxicity of TMAO is not to eat very much meat. Only about 1 in 4, a quarter of the population of bacteria, have the enzyme necessary to convert carnitine into TMAO. So if you're one of those people who does both, then you are more likely to generate TMAO, and therefore the downstream effects of epithelium tightening, inflammation, atherosclerosis, and so on. So it does, to some extent, matter what the bacteria are in your microbiome. Hypertension, and as I mentioned, the Western diet is a high fat, high salt, high sugar diet, and in the salt intake in a substantial part of people in this country, the salt intake is way above what it should be. Salt intake actually shifts the gut microbiome to Prevotella and Ruminococcus, cutting down on the relationship between that and lactobacillus. Lactobacillus is a stabilizer for Th17 cells. However, when their numbers are decreased, then so also is their effectiveness in cutting, maintaining blood pressure. Hypertensive humans have a linear relationship between their increased IL-17 levels and blood pressure. IL-17 decreases endothelial nitric oxide synthesis and increases vascular tone and aids in synthesizing or sensitizing to angiotensin II. So high salt intake plays a role, interestingly enough, significantly through the microbiome. Depression is a concomitant for many people who have diabetes, both type 1 and type 2, although it seems to be more common in type 2. And part of the reason for that, we think, is that dietary tryptophan, which we absorb from food, and to be honest with you, which is also synthesized to some extent by bacteria, are used to produce serotonin, a major neurotransmitter. And that serotonin level is established and has a balance in all of us based on tryptophan metabolism and on the gut microbiome. The microbiome has the ability to shift tryptophan metabolism over to the indole pathway, which in many parts of that are actually beneficial to us. But in addition, unfortunately, some of the bacteria have an enzyme system that involves dioxygenase enzyme that takes tryptophan and converts it through and into the kynurenine pathway. As a result, whenever the lactobacillus is decreased, lactobacillus is increased, what you see here is that the kynurenine pathway is stimulated. And what you want to do is to cut down that stimulation, allowing more tryptophan and therefore allowing more serotonin to be stimulated, and therefore potentially enhancing mood so that you ameliorate depression. Lactobacillus has been known as an antidepressant, and in fact is the foremost beginning bacterium to be studied in something called psychobiotics. For those of you who work in the neurology area, you'll know what I'm talking about. Dietary tryptophan is used for making serotonin. It can be metabolized through the kynurenine pathway. The dysbiosis associated with inflammation stimulates tryptophan to kynurenine. And what you want to try and do is to cut down on that stimulation. Lactobacillus inhibits that initial step in the kynurenine pathway, increasing the availability of tryptophan for serotonin production. Let's talk a little bit about how you nourish the microbiome, talk a little bit about fiber, one of my favorite topics. Many, whoops, that slipped back. There we go. Many of you are familiar with so-called prebiotics or prebiotic fiber. I think there's a concise definition here. All prebiotics are fiber, but not all fiber is prebiotic. That prebiotic essentially means that those fibers can be broken down to smaller and smaller substances that can then be used to make short chain fatty acids. So there are all kinds of so-called soluble fiber, fructans, polyglycosides, fructose oligosaccharides. You could go on and on. There are probably 30 different classes of fiber that can be substances that the gut bacteria will break down and that they will then help to stimulate the production of short chain fatty acids. Probiotics, there's a gazillion of them. Pretty much if you read the labels, and I would always encourage you to do that, you'll see that most probiotics actually contain pretty much the same families or populations of organisms. I call these, they've been around historically for many years. My nickname for these are the Amazon probiotics. You can get on Amazon and put in the word probiotic and go for a gazillion pages. Most of them contain relatively the same organisms. Only recently have some of them begun to list not only the organism itself, like Lactobacillus acidophilus, but also list the strain. We do know that different strains of the same organism do not behave the same. So you have to, the more you learn about these and the more the supplement manufacturers learn about them, the more likely they are to be listing the strain of the organism they are using as well. The ultimate food for the microbiome and for the newborn is essentially human breast milk. It contains prebiotics in the form of oligosaccharides, and it also contains lactose and other things for nutrition. And the bottom line is we do know that if you are keeping your microbiome healthy by ingesting human milk, you cut down on the likelihood that bad things will happen, such as type 1 diabetes. So you're calming the immune system with the ability to metabolize human milk. Whether or not you're exposed to it for a very long time, the fundamental is if you're not exposed to it, you increase the risk of developing type 1 diabetes. If you administer, exogenously administer probiotics, if you exogenously administer probiotics, you actually can cut down on the development of type 1 diabetes. And this is some results from the TEDdy trial, where a so-called Amazon probiotic was used. And you can see it decreases the risk of developing islet cell immunity significantly over the space of just a few years. Probiotics, prebiotics, and even fecal transplants are now being studied. This is a list of some of the trials that are going on, looking at their role in preventing diabetes and preventing type 1 diabetes in this case. But there are other studies that are looking at those same things in treating type 2 diabetes as well. So this is a great reference if you're interested in studies that are looking to prevent or cut down the development of type 1 diabetes. A shotgun approach, that is giving all the bacteria without actually knowing which ones are going to make a difference, has been tried in a number of diseases going back literally gazillions of years. The first fecal transplants were actually oral transplants of feces. The Chinese used them in 4000 BC. So it's not new, it's just the way we talk about it and the way we do it. Fundamentally, you can cut down the development of type 2 diabetes using fecal transplants. In these three panels, this is type 2 diabetes development in mice. This particular trial is in mice. A subsequent trial, quite an effective one actually, was done, published in 2021, using fecal microbiota transplants to stabilize residual beta cell function in people newly diagnosed with type 1 diabetes. That is, cut down on the immune response, quell the immune response a bit, and you cut down on the development of and moving forward with type 1 diabetes. For type 2, so-called Amazon probiotics have been used for many years. You'll see that they, in some cases, do have some effect in changing the development of insulin resistance and in changing actual fasting blood glucose. So there are studies with traditional probiotics that do have some modest effect. Here are measures of oxidative stress measured in the groups that took those probiotics. You'll see HSCRP dramatically decreased and GSH dramatically decreased as well. Most recently, a new era of probiotics, and those are what I call designer probiotics or medical probiotics. These are probiotics that don't necessarily use the traditional organisms, but use organisms that have been shown to have specific effects, such as those organisms that produce butyrate. In this particular study, a double-blind trial was done looking specifically at different mixtures of organisms that stimulate butyrate production and also organisms that stimulate mucin production. As you see in this study outcome, a blend of those organisms that are butyrate producers plus Akkermansia, those together had a significant effect. That's the green bars at the bottom of this slide. And this particular designer or designed or engineered medical probiotic is actually now commercially available. So the bottom line is we are moving forward on our understanding of probiotics and moving forward on our ability to actually tinker with them to make them better. So the bottom line is there's a lot we don't know about the microbiome. If you go to PubMed and enter the terms human gut microbiome, you'll find that there are nearly 25,000 articles that have already been published. So if you are interested in this area, interested in all of the different things, the relationships between microbiome and the brain, the gut-brain axis, microbiome and the lung, the gut-lung axis, microbiome and the bone, gut-bone axis. We have so many of these articles out there that we actually develop axes about all the different things that they do related to that specific human organ function. So a lot of reading to do, guys. And at this point, I will leave you with one final thought. When we try to pick out anything by itself, we find it hitched to everything else in the universe. Okay, so I'm ready if there are questions. I don't know whether Christy's still there or Megan. If you have any questions, make yourself available and let's see if we can answer them.
So far, no questions, but if anyone has one, feel free to put it in the chat. Starlin says, great job, Kim.
Thanks, Starlin.
Oh, you have a question here, Kim. Can you comment on the WFPD diet?
I don't have much information on it, so rather than comment, I am aware of it, but not as much of its effectiveness. There is a lot of studies on diet of different kinds, modified, Mediterranean, keto, I mean, a ton of them, but at this point in time, I don't know of any major trials specifically related to the effectiveness of that diet.
Does anyone else have any questions for Kim? All right, well, thank you everyone so much for joining today, and we hope you have a good weekend.
All right, thank you, Christy.
Thanks everyone. Thank you, Kim.
Thank you.